Okay, good morning, everyone. Thank you for joining. This is Gal from Almog Diagnostic. We're excited to have you here. Today we have a very special seminar ready for you to talk about the COVID-19 researcher toolbox and various ways to build your research system depending on the research study that you have. Our presenter today is Galia Abraham, who will be taking through the demonstration. So I hope you enjoy it. Please note you're all on mute. And below the screen, you have a Q&A section. Feel free to write your questions, and we will address them at the end, one by one by Galia. Thank you.
Hi, everyone. Good morning. This is Galia. So as Gal just mentioned, the webinar today is for the COVID-19 Research Toolbox. And I want to start with the first slide. This is to introduce to you to Almog team. This is how we looked in a normal day. And after the outbreak, this is how we are working. This is our new uniform and isolating immune cells from COVID patients. So before we're going to dive deep into the science, I'm going to highlight the seminar topics. First, I'm going to start with only 2 slides for scientific background. The background was already published and known in the internet. And then I'm going to present COVID Research Toolbox. We wanted to focus on 3 main topics, which are the neutralization agents. The first topic we're going to talk about it, then the immune response. And the last topic will be the pathway discovery. At the end, I'll be more than happy to answer questions. So, first of all, I'm going to talk about very short talk about the schematic structure of the SARS-CoV-2. The virus is around 100 nanometer in a diameter. It has the S protein, which is the spike that extends on the surface of the virus. And this spike protein is grabbed onto the human cell just before the invasion. Then this virus also have a structure of proteins, which are the N, the M, and the E protein, which moves inside the cells where they help the virus to form and stabilizes the RNA itself. The second slide is for the SARS-CoV virus invasion process itself. So first, it binds to at the host cell. It can be a lung cell, a kidney cell, and other known cells. When the virus spike protein launches onto an ACE2 receptor, a protease enzyme from the host side slices off the spike head. This cleavage releases the fusion mechanism, and the ACE2 receptor, I just want to note at this point that the ACE2 receptor, normally one of his function is to help regulate the blood pressure, which is a very important role inside the host body. Then the second part is the slipping inside of the virus. The virus and the host cell membrane are fusing, allowing the virus RNA, which is the molecule that encoded the genome. The genome of the virus is around 2 dozen genes. And now the virus is actually pouring into the cell body. There is a fusion between the membrane of the virus cell and the membrane of the host cell. A channel is being formed, allowing the N protein and the RNA to enter the lung cell or any other host infected cell. This slipping inside process taking about 10 minutes. Then there is the replication inside the infected cells. When the virus is inside the cell, it presents about 2 dozen genes, as I've mentioned, inside the cells, which translate into from the genes into the protein itself. The virus uses its own RNA copying machine, which is the polymerase of the viral, to make duplicates of the RNA inside the host cells. Some of the copies are utilized to make more viral proteins, such as the spike protein. The other one will actually package into a new virus particle, which breaks out of the host cells. Now, the fourth process that I didn't write here and showed you, but you can see here on the bottom, this is the breakout of the viruses from the cell host. And the breaking out is a vesicle carrying newly formed viruses that are merged with the cell membrane. One cell can release around 100 of virus copies to the microenvironment of the tissue. It typically dies because the resources inside has already used up by the virus, or it is being killed by one of the immune system cells. Some of the viruses head off to infect more cells in the environment, and the others will be exhaled into the air. Now, these two processes I've just described, the replication and the breaking out from the cells, are actually around 10 hours. Okay, so now we're gonna start to dive into, and we want to know what is your research focus. So we wanted to focus on three main topics. The topics are neutralization agents, the immune response, and the intracellular affected pathway. In each section, we want to cover and to answer two research questions. Our first research questions will be, does your agent neutralizing the cell-virus interaction? And the second question will be the characterization of this process. So when we're talking about neutralization agents, the research question that arises is, does my agent neutralizing this interaction? And we can actually divide it into two cell screening assays. One is the cell-free screening assay, and the second one is a cell-based screening assay. When we are talking about a cell-free screening assay, we're actually talking about two major components. The components are the spike protein, which is a recombinant protein form, and the ACE2. Now, the spike can come as a certain type of form. It can come either as only the RBD domain, or the spike domain, which consists of the S1 and the S2 subunits, and also as a trimer, which is also consists of the S1 and the S2 protein, which provides a more biologically relevant, which gives you a more site rather than the monomeric spike. Now, the actually thing that you want to consider before performing this screening assay is, what do you want to screen? Does your agent is a neutralizing antibody, or is it a small molecule? If it's a small molecule, then both screening assays can be good for you. But if you want to screen a neutralizing antibodies, then we recommend to use the spike plated, because one of the components has attached and fused to an FC. So it's only because of the fusion protein. Now, those two assay cell-free based detection methods can be either chemiluminescence or colorimetric, and your sample source can be serum, plasma, any drugs you wish to screen, or small molecules that you can design. So as for this is the cell-free screening assay. The second screening assays that can be performed are cell-based, which are mimic more the microenvironment inside the host cells. So here also we need these two components. One is the ACE2 components, which are expressed over the cell surface. And the other one is, instead of using the spike proteins, we are using a pseudovirus, which mimics the virus itself. Now, you can use either your cells of interest, and you can transfect or infect them with the lentiviral expressing ACE2, or you can use primary cells, such as pulmonary, cardiac, kidney, or endothelial cells. You can also use, if you doesn't want to prepare these cells, you can use a ready-to-use ACE2 expressing cells, and they come in three different forms: HEC2 and HeLa cells or CHO cells. Now, the lentiviral particle are actually the pseudovirus. They come in two forms. One is a GFP expressing particle, and the other one is a luciferase. Now, if you want to use the luciferase, it is recommended, since this is a very a very sensitive way to measure. You can see here the differences between a faint infection, a moderate infection, and a strong infection. Now, if you want to see and perform an imaging screening, then you can, of course, choose the GFP one, and you can see the image in the plate of your infected cells. So those are actually two kinds of cell-based screening assays, and covering the assays itself, the screening assay itself. Now, as you all know, in every assay that we are performing on the bench, we need controls. So we can recommend on two positive control. One can be serum or plasma from patient that has been recovered from COVID-19. They are all PCR positive confirmed samples. They are high and low titer for IgM or IgG levels. And each sample comes with a full serologically and clinical data on the patient itself. Now, if you don't want to use the serum or sample, you can choose to use the neutralizing recombinant antibodies themselves. There are recombinant antibodies also being isolated from a patient who've been recovered from COVID-19. They were isolated, identified, and then a recombinant protein was produced. Now, after you perform the screening assay, you actually answer the question, does my agent inhibits this interaction, the virus cell host interaction? So if you know, and the answer is yes, it inhibits, you want then to characterize this inhibition. So to characterizing antibody neutralizing inhibition, you can use a screening assay. The uniqueness of this assay is that this is a spike protein array. This array consists of 14 peptides of the spike, COV2, and 3 full proteins, which are the RBD protein, the S1 RBD, the S2 subunit full length, and the N protein full length. Each array consists of, one slide from the array consists of 16 samples. You can use, if you have more samples, more than one slide. And if you're using serum samples, then you need only 2 microliters from the original sample itself. Now, this was one part of the equation. The other part is the ACE2. If your agent is a molecule, a chemical molecule, you want to test its toxicity, because if it inhibits this interaction between the virus and the host cell, it might be toxic to the ACE2 receptor. You need to keep in mind that the ACE2 have a crucial role in the host body, and you wish to know it upfront before you're actually getting into clinical stages, if your molecule, if your chemical agent is safe and not toxic to use. So these are both assays that you can perform to characterize your interaction and your inhibition. So as for now, we covered the first topic, which was the neutralizing agent, and we answered both questions. Now the second topic will be the immune response related questions. For this topic is viral infection influence the immune system? Does it influence? How can we measure it? And using immunotherapy methodologies to study and target those things. Okay. Okay, so this is only one slide, only one introduction for the immune system defense measures. We have the innate immune system and we have the adoptive one. Both are going and work together. So when the infection begins, the innate immune system tries immediately to protect the host cells. The adaptive immune system gears up for a greater response. Now, as for the innate immune system, an infected cells releases interferon proteins that alert neighboring cells to create molecules that are trying to stop the virus particles from entering and reproducing. The interferon also beckon the cells, such as macrophages, in the bloodstream that can engulf the virus particles from penetrating and ring the host cells. Now, the adaptive immune system. The adaptive immune system, interferon, also alert B cells. They produce neutralizing antibodies that might recognize part of the spike protein and bind to it. And this is preventing the spike from grabbing onto a lung cell or any other host cell. Interferon also recruits the T cells, which can destroy viruses and also kill infected cells before the virus inside are bursting out of them. Now, some of the B and T cells will become eventually memory cells that can quickly identify and fight if there is a future invasion by the virus. So, when we're going to talk about the immune response, we have three major topics. We have the isolation of the target cells, we have the stimulation, and we have the analysis and the measurement for this stimulation. Now, it can be either performed by this order or the other way around, or any order you find and will be suitable for your experiment. So, as for the first topic, the cell isolation. Here, we want to actually distinguish between two kinds of samples. One, samples are from recovered patients. They can be either PBMC or plasma from recovered individual. They can also be COVID-specific T cells. Now, if your samples are infected samples and are very highly risk to use, then you need to consider a BSL-2 environment. So we want to present to you the MaxQuant titer sorter, which actually answer this type of samples. It is safe and sterile sorting to use. There are no tubing sets or fluidic inside this device. It's a very gentle, low pressure and fast sorting. Everything is actually happening inside this cartridge. And this sorter, like any other sorter in the market, it's fluorescence based. So as for the cell stimulation, if you're taking PBMCs from a recovered patient, you will need to reactivate those cells in order for them to produce cytokines. So those peptivators are actually pools of peptides for all virus protein. You can see here on the right side for the S, for the spike, for the S1, the S2, the N, and the M, there are available pools for this in order to stimulate the cells. And the peptides itself are 15 amino acids, each one long, and there is an overlapping of 11 amino acids between one peptide to another. Now, those peptides are already being used in clinical today when using it with patients that underwent bone marrow transplantation and now infected with virus. So other peptide pools are already being approved for GMP and clinical use, and the COVID-19 will be further on also being approved for this application as a GMP, a clinical use. Now, you want to perform a detection of this process. You actually isolated your cells and you stimulated them, and now you want to see the response. Now, the response is actually the cytokine secretion. So you can either enrich and detect or only detect those cytokines. So if you want to enrich and detect, you have the antigen-specific REST stimulation, and then you are labeling the cells. You have this two-headed antibody, which one side is attached to those cells, and the other side of the antibody is ready to catch the secreted cytokine from the cell because of the activation. Now, those secretion cytokines are captured by the antibody. Then there is a detection antibody labeled either with PE or ABC. And for the last step, there is an anti-PE or APC microbead, which grabs this entire complex and then isolates only those cells that are secreting. You need to take under consideration then when you are performing stimulation to PBMCs, not all cells will be stimulated and activated. So you want to isolate only the activated cells, and the isolation procedure will be based on the secretion of the cytokine. Here on the bottom, you can see the list of those cytokines that can be detected, and based on it, enrich the cells. Now, this procedure, I want to highlight the uniqueness. Is the entire procedure is a labeling of a cell surface markers and a cell surface secreting cytokine. Now, another secreting system that can be in order to identify those cells is calling the rapid cytokine inspector kit. Here you also restimulated your cells. After the stimulation, you're performing an inhibition. A Golgi inhibitor is added to the sample. So you're actually allowing the cells to accumulate the cytokines inside. In this kit, the staining is outer, cell surface staining, and also an inside staining. The inside stain is of those cytokines that has been accumulated. You can see here on the right, the working protocol, which consists only 6 simple steps. And here you can see an example for an analysis that has been performed on CD3 cells versus interferon gamma that has been detected. The upper row is from a healthy donor. The lower row is the recovered patient. And you can see that those cells, those are the control cells, unactivated, unstimulated. These cells has been stimulated with the N protein peptivator spool. This is with the M, this is with the S, and this is with the 3 pools. You can see here the secretion of the interferon gamma from the cell itself. And here you can see then when you are performing the enrichment of specific T cells, then you can see here also an example from healthy donor and recovered patient. This example is for TNF alpha, and you can see this is the control. This is upon stimulation with the N, M, and the S. You can see the stimulated and the secreted TNF alpha cytokine. This is a positive control of stimulation with another peptide pool, which is the influenza antigen. So as for now, we have been covered 2 of the main topics we wish to talk about, the neutralization agent and the immune response. And we're actually going to talk about the last topic, which is the intracellular affected pathway. So the very basic assay that can be performed in order to see those cytokine storm or any other cytokine or growth factor or anything you wish to investigate it upon this infection of the virus, then you can perform a cytokine array, which I'm sure you're all familiar with. It comes in 2 kinds of forms. There is a semi-quantitative or a quantitative array, and you can scan it and see all targets if you want a custom made, if you want something that's already pre-made or designed for a specific pathway that you can screen. Another way is to use the CRISPR-based solution. Now, the CRISPR-based can be harnessed to this research. How you can do it? You have 3 options. The first option is guide RNAs that already been designed for the SARS-CoV-2. And those are 15 optimized guides RNA that are from over 80 SARS-CoV-2 samples. So those guides are for the detection of the virus itself. It attached to the RNA of the virus. And then you can use the Cas13a, which is, if you compare it to the Cas9, it doesn't cleave and it doesn't bind to a DNA, but rather a double-stranded RNA, and it's performing only a detection. So if you're gonna add a few more steps, you can make it a diagnostic kit. Now, another 2 options you have is a SARS-CoV libraries. There are 2 libraries available. One library is of 330 human proteins that were collected from published and known data in the literature that known to interact. Those are human, the host proteins, and they are known to interact with the virus upon virus infection. The other library are 63 proteins, human proteins, that known to have drugs that inhibits them. So instead of using those drugs and perhaps suffer from side effects, you can use the CRISPR in order to inhibit those proteins. Now, after we've covered all three topics, I'm going to show you just one more last slide, which is a very broad range slide, because it actually gives you all kinds of additional essays and research tools that you can use that doesn't actually depend on immune or things that we were talked about. For example, if your samples are saliva-based, then there are arrays for saliva cytokine storm, and you can screen cytokines inside saliva from human patients. You can also perform live cell imaging with the, let's say, the lentiviral particle, which is the EGFP, when you can image the infected cells. You can also use kits that are fish probes, which actually, if you have your samples are tissue samples that were infected with the virus or cells that are already known to be infected, then you can actually detect the viral particle inside those cells. The fish probes will attach to them, and the fish probes are labeled fluorescently, so you can perform an imaging test. There are also signaling protein arrays, antibodies, and biosimilars. Biosimilars, I mean by drugs that are in a research grade and not in a clinical grade, so you can use them as inhibitors in your assays. Now, at this point, I want to thank you all for taking your time and joining us, and I will be more than happy to answer your questions.
Hello, everyone. Gali's, you can write question to Gali on your right side of the screen. There are a Q&A section. Please write them, and we will address them one by one. Thank you.
Okay. So one question that was just reading, do you distribute small molecules that inhibits ACE2, S1 interaction? Yes, we are. Isn't entry assay always superb to ELISA? I think so, because you can establish more data than you can establish with ELISA. When you're actually testing ELISA, then it's very quantitative and a very, very specific things that you are receiving from. When you're performing entry assays, you are actually mimicking what is actually happens inside the host cells. So you can see if you have this inhibition, non-inhibition, what is the effectiveness of your infection? How you can infect, how broad is the infection, okay? And as I've mentioned in the screening assay, there were two types of particles and invasion measurements. One was by GFP imaging, and the other one was a luciferase, which is even more sensitive because you can even detect a faint invasion. Okay. Which one? Some companies have them in an inhibitor kit, but they do not sell them separately. Okay, if you want, we will be happy if you can contact us, And I will give you an entire list of inhibitors that doesn't have any correlation with kits, but actually sells as standalone molecules. You're welcome, Shira. Are there any more questions? Okay, so if you will have
okay, we can see that
Antonina. Hi.
Antonina, do you mean to ask a question, or it's just saying hello? Please write to us in the Q&A section. Oh.
Okay, do you have mouse peptides? What do you mean by mouse peptides? Yeah, oh, I understood. You are actually referring to the peptivators, right? Did I understood you correctly, Antonina? Do you have peptides to mimic COVID-19 in mice model? Well, I wish to, okay. Our peptivators are actually the virus itself. So if the virus are being recognized by the mouse immune cells, then they will be helpful for you. I do not know if mice are actually know this virus or the virus can infect them. If yes, then you can stimulate it using these peptivators, of course. Okay, thank you. Yechezkel, thank you. And Yuval, does your neutralizing test can replace neutralizing assays with live virus? Yes, the pseudovirus actually mimics a live virus. Now you still have to use the BSL-2 facility because although this virus doesn't replicate itself because its lack of the signal of replication, still you are working with the virus, but it's not the real COVID-19 virus, okay, with all the risks that came along, but rather a pseudovirus, which is the HIV backbone, and instead of the VZV gene, you have the spike protein. So you're actually mimicking the invasion and the infection. Okay, a question for Meital Galtanami. Hi, Meital. Do you have an assay with the live viruses? What is the titer of your pseudovirus? Okay, so the titer actually vary from lot to lot. I can check it and send it to you, okay? What are the titers that we have now? And these lentiviral particles are actually mimics live viruses. So we don't have the COVID-19 viruses because we are not isolating those viruses from patients. Okay, hi Miriam. Miran, sorry, Miran. Do peptivators stimulated macrophages instead of LPS? Well, the peptivators usually stimulate T cells. Macrophages are usually stimulated by LPS or interferon. I can check it if you want to. Thank you, Meital. Okay, so thank you all for being and listening to this webinar. If you have any more additional questions, please kindly contact us. We will be happy to assist you. Have a nice day.